This part of the lecture is about the different types of neurons and the neuroglia, and it also starts to talk about the action potential. So here, just showing the different types of neurons. That picture that we saw previously of the neuron and its parts was actually showing a multipolar neuron. So there are more multipolar neurons, and so this is what the first picture is showing. 99% of neurons look like this. So they have the dendrites coming out, and they have the axon. So this neuron is called a multipolar neuron because there are multiple branches coming out of the neuron. But yes, these are most of the neurons of the central nervous system. Now the other two are more rare, bipolar neurons. You're actually gonna learn about this in A&P2 when you look at the eye and the ear. Bipolar neurons are in those special senses regions, also the olfactory nerve. So this is the second picture here. They call this a bipolar because only two branches come out of it. So the top part here, this is then going to lead to the dendrites, and the bottom part is the axon, and then the axon is going to send the information to the other neurons. Then the unipolar neuron. So this is called a unipolar because only one branch comes out of it, then that branch separates into two. Yeah, so it kind of looks like it's got a neck and then two arms out of it. And so these are the sensory neurons. So just be able to recognize these different pictures. Okay. So then the classification of neurons, this is really based on the function of neurons. And so sensory neurons are usually unipolar, some are bipolar. So sensory neurons, oh here, the other word for sensory is afferent. What this means is that the information comes in. So it brings in the information to the central nervous system. So the sensory neurons, think again for the special senses. It could be, these are transmitting the sight information, the taste, hearing, touch. And also in this diagram, sensory receptor just means the part of the neuron that picks up the information. And so then the neuron then sends the information to the central nervous system. In the central nervous system, the neurons just, as collectively, they're just called interneurons. And so for the interneurons, just know that these are the neurons of the central nervous system. So the neurons in the brain and the spinal cord. Just a general name for them. And then the motor neurons, they're multipolar. And so the motor neurons then take the information from the brain and spinal cord and then send it to the muscles, the organs. Okay, so now next, the neuroglia. These are the helper cells. And so they provide structural supports. When we are developing embryo fetus, these neuroglia actually help to move the neurons to where they're supposed to go. Really interesting. They release hormones, growth factors to nourish the neurons, help them to grow, and also aid in the formation of synapses, meaning the connection between different neurons. Okay, so here are the different types of neuroglia. Oh, and make sure you know that these are the central nervous system helper cells, these four that are listed here. Astrocytes, these provide structural support. They help to move the young neurons, so when the brain is developing, to move the neurons where they're supposed to go. But especially know these are for the structural support. They're helping to keep the neurons in place. Oligodendrocytes, these make myelin for the central nervous system. Schwann cells are instead in the peripheral nervous system. Microglia, know that these are immune cells. These are eating the bacteria, parasites, killing the viruses, also somewhat of a structural support too. Then the ependyma, or the ependymal cells. These are lining the spinal cord and the brain, and what they do is they filter the cerebrospinal fluid. Okay, so what this means is I'm going to try to draw a picture here. So let's pretend that this is the brain, or it could be the spinal cord, so either one. So right on the surface of the brain and the spinal cord are these cells. So I'll try to make it a little bit larger here. Okay, so just pretend that these are cells here. And so these are the ependymal cells on the surface. And then around our brain, around our spinal cord, we have this fluid called the cerebrospinal fluid, CSF. This fluid contains nutrients, it contains growth factors, but it could also contain some toxins, some waste products. So you don't want that bad stuff to get to the brain. You only want the nutrients, the oxygen, the ions to get through. So these ependymal cells are filtering this fluid so that only the good stuff comes in. Okay, yeah, so this picture is just kind of showing all these neuroglia of the central nervous system. Oligodendrocytes, these are making the myelin sheath. So that's the yellow stuff that you're seeing that's sitting on top of the axons. Microglial cells, these are the immune cells. They're going to eat the bacteria, the parasites. Astrocytes, these are providing the structural support. They're keeping the neurons in place. And then over here, kind of what I tried to draw on the previous slide, showing the ependymal cells. So here, think this is supposed to be the brain. The ependymal cells are on the surface of the brain and the spinal cord, and they're making sure that only the good stuff comes into the brain or the spinal cord, not the bad waste products or the toxins. The helper cells for the peripheral nervous system, there are only two types here. The Schwann cells. Schwann cells are like the oligodendrocytes. So we'll say here similar to oligodendrocytes. And they're making the myelin sheath. Myelin sheath, we're going to learn, speeds up the electrical signal. Speeds up the speed of nervous signals. Then the other one, the satellite cells. These are similar to the astrocytes. So they also provide the structural support. Keep the neurons in place. Now, both the Schwann cells and the oligodendrocytes are making the myelin sheath, but there is a very big difference between the two. So the Schwann cells, once again, they're in the peripheral nervous system, the nerves in your arms or your legs. They help to guide growing axons. They secrete growth factors, but they also help to repair neurons. Okay, so if you have an injury, let's say in your arm, in your nerve gets damaged as well, the Schwann cells are actually going to help to repair it by releasing these growth factors. Oligodendrocytes, they're in the central nervous system. They also make the myelin sheath, but they actually release anti-growth factors and they prevent repair. So this is why repairing the neurons in the central nervous system is very hard. In children's brains are more plastic, they're able to repair themselves, but oligodendrocytes are going to stop that. Now, the reason for this is that you don't want the brain to keep growing, you don't want the spinal cord to keep growing, you want it to stop, right, after the growth is finished. And so this is a reason for them releasing these anti-growth factors, but of course as a result, this is having a detrimental effect because the central nervous system. Okay, so now next we're going to start to go into the electrical signals. Synapse. Synapse just means where the neurons communicate with each other. And so it's where the neurons are touching each other. So what we're seeing is not the best picture, but these are supposed to be two neurons. In this case, the two neurons, here's the presynaptic cell or the presynaptic neuron. This is the neuron that sends the impulse. So this is going to be the first neuron. And the postsynaptic neuron or the postsynaptic cell is the second one. And so the connection between the two is called a synapse. The synaptic cleft is actually the space between the two. Space between the two neurons. Okay, so what we're going to see with the synaptic transmission, meaning how the information goes from one neuron to the next, is a one-way transfer. So meaning that the first neuron is going to release neurotransmitters, and this is going to go to the second neuron. And so this can continue down to the next, to the next, to the next. So there might be many, many neurons that are in this line. And so the neuron that releases the information would be the presynaptic, and the next neuron is called the postsynaptic. Yeah, so what we're going to learn is the electrical signal goes down the presynaptic neuron. This electrical signal is going to cause the release of neurotransmitters. So if you remember, the ends of the axons are called the synaptic knobs. This is where the neurotransmitters are stored. And so when the electrical signal reaches the synaptic knobs, then the neurotransmitters get released, and the neurotransmitters then go to the next neuron. And let me also just review this. The Nissl bodies, the rough ER in the cell body, this is where the neurotransmitters are made. Neurotransmitters also need to travel down the axon, and then they go and wait in the synaptic knobs. Okay, so they're made in the Nissl bodies, they're stored in the synaptic knobs, and so when the electrical signal comes to the synaptic knobs, then the synaptic knobs release the neurotransmitters. Okay, so this is what this second part is saying. Impulse travels down the axon of the presynaptic neuron to the axon terminal. Again, the other name for the axon terminal is the synaptic knob. Synaptic knobs. When the electrical signal comes down, this is going to cause first, so there's a step in between. First, calcium has to enter the synaptic knobs. Then the neurotransmitters can get released. Okay, so we're going to talk about this again, and I'm going to show you the video that will help you to visualize this. First, calcium has to go in to the synaptic knobs, then the neurotransmitters get released from the synaptic knobs, and the neurotransmitters are sitting inside these vesicles. So what this picture is showing here, it says here the presynaptic neuron. So this is showing the end of the presynaptic neuron, the synaptic knobs, and these little bubbles. There are also, other organelles here too. But so these vesicles contain the neurotransmitters. First, the action potential runs down. This electrical signal runs down. Calcium has to enter first. And then after calcium enters, neurotransmitters can get released. And here it's showing Ca plus 2 for calcium. The plus 2 is for the electrons that are lost by calcium, because calcium tends to lose its electrons. It has those 2 extra electrons. Okay, and the next thing here, cell membrane potential. What this really means is that the inside of the cell, and this is true for any cell, not just for neurons, the inside of the cell is always more negative when the cell is resting, and the outside of the cell is more positive. Okay, so this is also called the polarity of the cell membrane, and this is due to the unequal distribution of ions. Okay, so the inside of the cell has a lot of negatively charged ions, negatively charged proteins, and so the inside of the cell is more negative.